Hi everyone, we are Team 1 and we're going to be presenting on umami receptors. And in order to do that, we've roughly outlined the flow for today. So we're first going to start with some basics and then we're going to define umami and what it really is. And then we'll get into the actual fun part, which is the umami receptor specific information. So basics of anatomy physiology of tasting sensation is represented here. So on the image on the left, you can see on the tongue, there are all these little dots represented. Those dots, which you can also probably see in the mirror on your actual tongue, represent papillae. Each papillae contains taste buds, which are represented here in the red lines. And taste buds are the anatomical pores that sense taste. In the taste bud, the gustatory receptor cells use villi, the taste hairs, to sense different chemicals in the foodstuff or tastins, and then it will pass the signal on to the gustatory sensory neurons. And in order for the chemicals in foodstuff to be sensed by the hairs, the foodstuff or the tastins must be first dissolved in saliva. So as we zoom into one specific taste bud, that image is represented here. This is inside of one taste bud. We can see on the right side, we see the microvilli and villi, which are those hair-like structures that will actually bring in the input from the tastant. And then within the buds, we see the taste receptors, and these are connected to the afferent nerves, which are represented here by the yellow lines. And those afferent nerves send signals to the tongue, from the tongue to the brain. Yeah, and that's all I want to say about that. So that's kind of the basic process. And here's kind of one overall image that can represent that. This is kind of the general process of taste reception. So the tastins are dissolved in the saliva. They'll bind to the taste receptor on the gustatory cell via the villi or the hair fibers. And then that taste receptor, which is thought to be T1R1 and T1R3, will do its magic, and that cell will depolarize, send the signal through the cranial nerves 7, 9, and 10, through the thalamus, into the gustatory cortex of the brain. And I think most of us grew up when science acknowledged the 4 primary tastes being bitter, sweet, sour, and salty. And umami was not on that list. It wasn't on that list when I was growing up, but it has been recently acknowledged by the West. It's kind of what is now acknowledged as the 5th major taste that the tongue can have. And what umami really is, so it can be found in savory tasting food. It contains L-glutamate, which is most commonly found in protein-rich foods because glutamate is an essential amino acid. That means the body doesn't make it and it needs to be ingested. And interestingly, if you don't already know, MSG is a variation of this. That's why like MSG is so tasty whenever it's added to food. And what it is, it stands for monosodium glutamate. So it's just the amino acid glutamate bound to a single salt, and it makes it like a billion times tastier. So now let's actually talk about the specific umami receptor business. These 2 images are kind of representing 2 different visualizations of the same process. And the one thing I want to say before we actually move into talking about the specific way that this travels through to have a signal to the brain is that it's thought that the receptors have relatively low binding affinity to the glutamate as kind of a practical feature because glutamate is found in high levels in food. And so if there was a high binding affinity, then your mouth would just be like on steroids in terms of that glutamate binding and sending signals and stuff. And so that's kind of an interesting thing that I didn't know about. So as the food stuff or the tastant enters the mouth, and I'm going to actually show you guys this on both images just because I think they're both helpful in their own way. But let's first go with this blue one here. So this is the food stuff entering the mouth. It's represented here by the diamond. So let's just say you have like soy sauce or something that contains glutamate that is thought to be bound to be going into this process. So it's this yellow diamond here on the right. This is going to bind to GPCR that is represented here, and that is the G protein coupled receptor. These are the primary receptors of interest, and particularly it's thought to be a shorter form of M-GLUR4. metabotropic glutamate receptor 4. So this is thought to just be a shorter version of that. When it binds to that receptor, the alpha-containing subunit shown here is activated, and this acts by activating the phospholipase C, which is represented here, the PLC beta 2. So whenever this is activated, what this does is it breaks down this thing called PIP2, and it breaks it down into two things. It breaks it down into DAG, diacylglycerol, and IP3, IP3, which is inositol triphosphate. So we can see that here. So here's the DAG molecule coming out, and here's the IP3 coming out. The IP3 specifically travels to the smooth endoplasmic reticulum. We can see that here. And it causes calcium to exit the smooth endoplasmic reticulum. And this is kind of the key feature for translating that umami taste into a neural signal. Because whenever calcium exits, it will open the TRP5, which functionally results in a release of ATP. And this acts to send an action potential through the afferent neuron to the brain center. And then it will move through the thalamus, and it will make it to the gustatory cortex. And specifically, this is going to move through cranial nerve 7. And something that might be kind of a fun fact about cranial nerve 7 is that the facial nerve, the cranial nerve 7, innervates the front 2/3 of the tongue, which again consists of those umami receptors. So in this entire process, also just kind of an added thing that we need to mention is that we see an overall decrease of cyclic adenosine monophosphate, CAMP, which is a derivative of ATP intracellularly, because again, that ATP molecule is thought to act as that messenger to send the signal through the afferent neuron. So all of that same information is also represented here. So the food is this little, this fire molecule. It moves in, it binds to GPCR, that moves out. Oh gosh. Okay. And this, again, is thought to be that shorter form of mGlu-R4. And then this binds, this releases the alpha-containing subunit, and that activates the phospholipase C, which turns PIP2 into those two compounds. We're really only interested in IP3. IP3 goes to the smooth endoplasmic reticulum and releases calcium. And that really is, like I said, the interesting portion of the entire process. That's what translates it into a neural signal. Sends ATP, and ATP is going out, getting to that afferent neuron and sending the signal through. So why do we care about this at all? Why do these actually happen this way, and why do we care? So umami, I think everybody might know this, but I hope everybody knows this, that umami is like the mother of all tastes. It's super delicious. It's like partially addictive. I mean, it's not actually addictive, but it's really, really good. And your tongue definitely wants more of it all the time, I think. And what this ends up doing is it helps humans increase their salivary output. So we have a lot of salivary output in response to umami taste. In fact, it provides more sustained salivary output than even a sour taste would. And I think sour is typically the most, we think that we have more salivary input with sour things, but apparently sour taste only gives us short spikes of saliva, but the umami gives us sustained activity. And saliva is very important biologically for digestive aids, as well as oral hygiene. So it's necessary for both of those things. Your food is, some of your food in your mouth, whenever you're chewing on it and it's mixing with saliva, will actually begin the process of digestion in the mouth with saliva on certain foodstuffs. And I found this kind of random quote that I think also adds to the functional importance of this, which is that umami taste receptors are thought to exist in the gut as well, which means that they may also play a part in digestion. So we know that having more saliva in your mouth can increase digestion because it's starting the process in your mouth, but that was really interesting to us. So that's all we got for you today. Thank you so much for paying attention.